Assalamualaikum and good morning. I'm Puan Maznah. Will be explained for the video 2 for subdivision for subtopic meiosis. For this subtopic, your course learning outcome, you should define the chromatid, homologous chromosome, synapsis, bivalent, tetrad, chiasma, crossing over, and centromere. And also, you can state the stages of meiosis 1 and meiosis 2. It's easy for you for this terminology. We starting with the chromatid, sister chromatid, centromere, homologous chromosome, bivalent, tetrad, synapsis, crossing over, and last with the chiasma. Now we go on the first term, chromatid. Chromatid, that means one of the two identical half of the duplicated chromosome, and for the sister chromatid, that means two copies of duplicated chromosome attached to each other by proteins at the centromere. So that means here, if the yellow color, that means sister chromatid, and the blue, that means chromatid. If joined together, we refer as a sister chromatid. And this first chromatid will be separate during anaphase, mitosis, and anaphase 2 for second meiotic division. We go on the term centromere. Centromere, that means centralized region of the joining of the two sister chromatid. So here they are centromere, and each of the sister chromatid have the kinetochore, a structure protein associated with the specific section of the chromosome DNA at the centromere. The kinetochore will be join the two sister chromatid face of the opposite direction. And function of the kinetochore as a attachment site for microtubule necessary to separate during cell division. Okay, meiosis, they are differ compared to the mitosis. Why? Because they have two cycle of nuclear division. We refer as a meiosis 1 and meiosis 2. So meiosis 1, they have conceived 4 phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. After telophase 1, they are followed by cytokinesis to form 2 haploid daughter cell. And meiosis 2, they are similar to mitosis, and we have 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. And this follow also by cytokinesis to form 4 haploid daughter cell. Doesn't have DNA repopulation between these two divisions. Okay, the important part for the prophase 1, we have another term that means like homologous chromosome, sister chromatid, or four sister chromatid, or bivalent, or tetrad. So this diagram refer to this kind of the terminologies. Okay, now we go on another term, homologous chromosome. A pair of chromosomes of the same length, centromere position, and staining pattern that possesses gene for the same characters at the corresponding loci. So because we have the pair, we have two. The orange color, that means they are from the father, we refer as a paternal chromosome, and the maroon color we refer as the maternal chromosome, that means it's coming from the mother. And both chromosomes have have carry their gene, the same inherited character. Okay, another term, bivalent. Bivalent, that means a pair of homologous chromosomes that line up beside each other. So if beside, we refer as a bivalent. And from that also we have another term, tetrad. Tetrad, that means a pair of homologous chromosomes with 4 chromatid. We have 2 important events occur in prophase 1. The first, synapsis. Homologous chromosomes come together or pair up of homologous chromosome. So the result we refer as a bivalent. So that means synapsis, they are pair up of the homologous chromosome. Let's see in the diagram the structure to form tetrad and also the word synapsis. So homologous chromosome, we have sister chromatid, and if the pair up of the homologous chromosome, we refer as the tetrad. The process, synapsis. The second event occur at process 1, crossing over. Exchange of genetic material between 2 non-sister chromatid of homologous chromosome or bivalent. So here, the segment of the non-sister chromatid. The segment of the non-sister chromatid will be break off and recombine. So that means at the end of the prophase 1, we have the genetic variation or genetic recombination. So that means sister chromatid is no longer genetically identical. And the last one, chiasma. Chiasma, region at which crossing over occurs between 2 non-sister chromatids. So that means pair of the homologous chromosome remain in contact as they begin to separate to form cross shape or egg shape. If many, we refer as a chiasmata. Okay. So we end of this video.